My name is Dr. Teresa Duda. I work with Dr. Sharma as a member of his research team. It's a pleasure to continue talking to Dr. Sharma, the retired distinguished professor of biochemistry and molecular biology. Just as a brief reminder, Dr. Sharma, together with his group and collaborators, authored 303 publications, including 3 books, 20 monographs, and held 2 US patents. His research was awarded for $7.3 million by the numerous awarding agencies, mostly National Institute of Health, National Science Foundation, Pennsylvania Lions Club Eye Research Foundation, Damon Runyon-Walter Winchell Research Fund, and Veterans Administration Research. He is the discoverer of multi-limbed membrane guanylate cyclase molecule. It took him 54 years from 1969 to 2023 to accomplish this colossal work. The discovery is monumental in the field of cell biology. With a human touch, the present video recounts the history of the foundational LIMP-1 of the adrenocorticotropic hormone ACTH modulated signal transduction pathway. Dr. Sharma, you finished your previous narration with the statement that after receiving 2 Veterans Affairs Hospital grants, you were able to build your own research program and your research group as independent research program. Could you please tell us about the beginning?
Yes, Mrs. Linda Sutcliffe was the first member of my group. She joined as a full-time technician with the degree of Bachelor in Science from the University of Georgia, and also being a member of the cheerleader group of the football team. She was extremely present and eager to learn the new tools of biological research. In the beginning, she chiefly helped me to do day-to-day experiments. Then she quickly progressed, learned how to prepare and deal with the isolated cell preparations, how to carry out the incubation experiments, and assay the corticosterone, cortisol levels. She became an extremely valuable asset, and I was happy to have her assist me. My research advanced significantly, resulting in 3 major scientific publications. In line with my vision and official duties to develop a research program centered on the theme of elucidation of the control mechanisms of the hormones at the submolecular level, I chose to investigate the action of the adrenocorticotropic hormone, abbreviated as ACTH, a polypeptide structure. In contrast to these steroid hormones secreted in the pituitary gland, ACTH, by targeting adrenal glands, controls the production of another hormone, cortisol in animals and corticosterone in lower animals, rat. Cortisol is a key element in responding to stress, fighting infection, and maintaining blood pressure. It is generated by the fascicular cells as shown in the figure. Thereby pituitary gland and adrenal gland bond.
Why did you choose this model to enter the field of hormones?
It was for 4 reasons. 1, it was a basic science problem, yet had a direct clinical relevance. Two, it was in line with my prior experience with the plant steroids. The envisioned hypothesis was that the pivotal molecule in both was the biosynthesis of cholesterol. Yet, in animals, only ACTH is active because plants do not embody the pituitary gland. Three, the clinical steroid biochemistry core laboratory was already and routinely involved in testing the levels of ACTH in the patients. And finally, the fourth, Dr. Abbas Kitabchi, VA hospital chief, was highly supportive of this choice and wanted my help in his clinical application of this research. This course satisfactorily fulfilled my official duties both in the VA hospital and in the basic biochemistry department. Yet, I had to raise the funds for my basic research. In sequential many steps, the foundation of the ACTH modulated steroid hormone research project started to shape. Its accomplishment was the historic scientific event in the generation of the cellular signal transduction field. The field demonstrated that the ongoing dogma of the cyclic AMP being the universal hormonal second messenger is flawed. For this concept, Dr. Earl Sutherland Jr. had won the 1971 Nobel Prize in Physiology. Consequently, the prevailing thought was so intent that the sole mechanism by which all hormones exhibit their physiological activity is via their common second messenger, cyclic AMP. This meant that structurally the three-component GTP binding protein was the trigger of the ACTH modulated transformation of the cholesterol to cortisol corticosterone synthesis by the fasciculator cells in the adrenal gland. It is shown in figure 1. Following Dr. Sutherland, 2 additional Nobel Prizes in physiology were awarded for this concept to Drs. Alfred J. Gilman and Martin Rodberg in the year 1994. I have discussed the issue in video 3. Wow. Notably, at the time, this possibility was not even remotely considered. Thus, it was a game-changing concept. Retrospectively, it constituted a major event in the symposium held in the Second World Congress on Biologically Active Peptides, Waldorf Astoria, New York, on May 17 to 21, 1987, where I was the plenary speaker. Our group, constituting one technician, Linda Sutcliffe, and I, with resources of mere $41,000 from the VA hospital, challenged this exclusive dogma and undertook this Herculean task.
Thus, you wanted to verify, proof or disprove whether cyclic AMP has a role in ACTH signaling. How did you approach this goal experimentally?
Sequentially, we demonstrated 1. The dogma is not applicable to the ACTH modulated steroidogenesis in the fasciculator cell of the adrenal gland, and 2. Further showed that there the strong possibility exists for the cyclic GMP to be the physiological mediator. This task was successfully completed from the year 1971 to 1973. Then the financial resources started to pour in and continued into the year 1974 and then peaked to the year 2023 when the task was brought to completion. The task begins. The first critical question was, is cyclic AMP the physiological second messenger of ACTH? It necessitated the development of new technology. Paramount was the bioassay that detects the physiological concentration 0.5 to 10 microunit of ACTH in the human plasma. Second, the isolated system of the fasciculator cells. The animal model was rat. Both tasks were accomplished as follows. The adrenal gland was treated with trypsin to selectively remove its zona glomerulosa and zona reticulosa, yielding the sole region of fasciculator cells. This in turn served as a source of the pure isolated cells used in the experiments. The cells were physiologically pure, solely synthesizing corticosterone, the counterpart of the human cortisol, in response to as little as 0.5 microunit of ACTH. Conspicuously, there was a small lag period in kicking on the response to ACTH, demonstrating the auxiliary role of a labile factor. The factor was a protein because its inhibitor cyclohexamate and not actinomycin blocked the lag. Demonstrating specificity, these fasciculator cells did not respond to other polypeptide hormones like insulin, growth hormone, proinsulin, glucagon, and epinephrine. In further affirmation, one, there was a linear relationship between the number of cells, 50 to 400,000, and steroidogenic response to ACTH. Number two, in time dependency, 0 to 180 minute interval, the maximal physiological concentration of ACTH, 50 microunits, generated corticosterone production in a linear fashion between 6 and 180 minutes. Number three, a definite short lag period, 6 minutes, occurred before the commencement of the linearity in steroidogenesis, a repeatable phenomena with as low as 5 microunit concentration of ACTH. The physiological purity of the cells was matched with their morphological integrity. The light and electron microscopic analysis demonstrated that the cells embodied the typical fasciculator cell attributes, a vascular mitochondria, the excessive cellular content of lipid globules, endoplasmocrine, a secretion of lipid globules together with the smooth surface endoplasmic reticulum, all characteristics of the pure fasciculator cell.
It's amazingly impressive that you were able to develop these delicate technology tools with this rat model. Were they applicable to human model as well?
Yes, the applicability of the isolated fasciculator cell assay system in a human study established that the plasma ACTH level in the fasting normal subjects is 2 microunits plus minus 0.1 microunit. The value increases about 6-fold when the generation of cortisol is blocked through the administration of mitaropone, a steroidogenic inhibitor. Thus, we had an extra sensitive and historical tool in our possession. We now could detect the physiological concentration of ACTH, and these could be used in vitro constitution studies for direct elucidation of the molecular mechanism through which ACTH controls the steroid hormone cortisol. In turn, we are now well positioned to systematically define the ACTH-dependent physiological process of stress, fighting infection, and maintaining blood pressure. This was a critical technological advancement to understanding the disease processes of stress and how to conquer them. In addition, and importantly, we could apply this knowledge to understanding abnormalities in the 494 endocortical carcinoma cells of rat. These had recently been developed in our laboratory, and later the patent was awarded to me in the University of Tennessee for the detection of this cancer in the patients. Cognizant with the prevailing concept that all hormonal actions occur through the cyclic AMP second messenger system, before the year 2002, this possibility was also considered in our studies. If applicable, the transformation of the ACTH signal into the production of the steroid hormones would be a three-component system consisting of the receptor, the G protein, and the adenylate cyclase. I have discussed this issue in video 3. Structurally, the adenylate cyclase is composed of two cytoplasmic domains and two membrane-spanning domains, each of which contains 6 transmembrane spans. As will become clear, this was not the case. Thus, the ACTH hormonal signal transduction system was unique physiologically, biochemically, and molecularly. Notably, this conclusion was arrived at through experiments that were conducted under conditions that were almost identical to the physiological conditions. The 500,000 fasciculata cells representing one rat adrenal gland or its counterpart, the endocortical carcinoma 494, were used in the reconstitution studies. The findings of these were unique as follows. ACTH stimulates the biosynthetic step of steroidogenesis from the 20S20 hydroxycholesterol. This step is dependent on protein synthesis and is rate limiting. Yet the cyclic AMP does not mimic this stimulatory action of ACTH. Thus, cyclic AMP is not the physiological second messenger of ACTH, as was thought previously. This negated and challenged the universality of the cyclic AMP second messenger concept. Accordingly, in an important advancement to the hormonal cancer field, ACTH in the carcinoma cells inhibits steroidogenesis, yet cyclic AMP does not. In a bell-shaped curve, the physiological concentration of ACTH, 2.5 to 10 microunits, does not raise the level of cyclic AMP, but does stimulate steroidogenesis. Notably, in another remarkable finding, ACTH stimulates the peak synthesis of cyclic AMP with a concomitant rise in corticosterone. Thus, it was concluded that cyclic AMP, not the cyclic AMP, is the physiological mediator of the ACTH hormone. This was the beginning of a new cell signaling transduction pathway. A surprising finding was that the supraphysiological concentrations, 10 to 100 microunits of ACTH, indeed stimulate cyclic AMP levels in a concentration-dependent manner with a concomitant rise of the corticosterone level. Notably, under these conditions, there was no rise in the cyclic GMP levels. They later established ACTH second messenger. Taken together, these studies established that the physiological second messenger of ACTH is a cyclic GMP, thus negating the concept that cyclic AMP is a universal second messenger hormonal action. Also, they demonstrated that cyclic AMP is only a pharmacological transmitter of the ACTH response. A mathematical equation was developed where cyclic GMP was a physiological messenger and the cyclic AMP the pharmacological messenger of the ACTH-mediated process of steroidogenesis. This equation is shown below, and where in this equation S and A represent the concentrations of GTP and ACTH respectively, and K1 and K2 equilibrium constants. This demonstrated that the enzyme combines with the second molecule of ACTH with greater affinity than the first, and there is allosteric modification of the structure by the first molecule of the hormone bound.
These achievements are indeed admirable, not only to the hormonal field of ACTH signaling and ACTH-mediated steroidogenesis, but also are applicable in general to understanding of cancer, starting a new era of signaling pathway where membrane guanylate cyclase generated cyclic GMP is a vital component. Would you please elaborate more on this subject?
Yes. Systematically guided by the template, which is the figure shown below, we started to define morphological and molecular attributes of the isolated fasciculata cell and use it to deepen our understanding in the biochemistry of the ACTH-dependent hormonal steroidogenic system, and notably began to pursue in defining the membrane guanylate cyclase cellular signal transduction pathway at the molecular and physiological terms. Summary of the concluded results is the light and electron microscopic analysis demonstrated that the isolated fasciculata cells were morphologically pure. Accordingly, there was a linear relationship between the number of cells, 50 to 400,000, and steroidogenic response to ACTH. 2. The time dependency study showed that in 0 to 180 minute time interval, the maximum physiological concentration of ACTH, 50 microunits, generated corticosterone production in a linear fashion between 5 to 180 minutes. Number 3, notably, there was a definite short lag period, about 60 minutes, before commencement of the linearity in steroidogenesis. This phenomenon was repeatable with 5 microunits of ACTH. This event combined with the fact that number 5, cyclohexamide, yet not actinomycin D, inhibited ACTH-dependent steroidogenesis, indicated that ACTH targeted the fasciculata cell via labile synthesis of a protein molecule. A finding of extraordinary importance that paved the way for future research was that isolated fasciculata cells beside ACTH were also responsive to the cyclic AMP and also to guanosine cyclic GMP, as well as its derivative dibutyryl cyclic AMP and the process of steroidogenesis. Yet their non-cyclic ribonucleotide 5' phosphate derivatives were ineffective. Thus, the cyclic nature of these molecules were the effective factors in the process of steroidogenesis. Number 7 finding was it was a follow-on study demonstrated that these cyclic nucleotides act independently of ACTH, and one or all of them are the mediators of ACTH. Because, in contrast to ACTH, they were generated intracellularly, one or all of them were the intracellular mediators of ACTH. To solve that mystery, it was important to determine that the isolated fasciculata cell of the adrenal does not contain other factors that affect the intracellular regenerated level of the cyclic nucleotides. Relevant to this reasoning was at this parallel study of Dr. George Chung which showed that mammalian cells contain phosphodiesterase activator that hydrolyzes the intracellular generated cyclic nucleotides, making them biologically ineffective. Yet, analysis established that the fasciculata cell has no phosphodiesterase activity. Thus, the steroidogenic activity of the cyclic nucleotides was authentic. And one or more of them was indeed the ACTH mediator of the steroidogenesis. The cell was extraordinarily tailored to decode the biochemistry of the individual step involved in the ACTH-dependent steroidogenesis.
You mentioned earlier that you also did experiment with the isolated adrenocortical carcinoma 494 cell line. How did these two completely different cell lines help you in achieving your overall goal?
To apply this basic knowledge of the phenotypic characteristics of the fasciculata cell, its changes accompanying its transformation to the cancer cell adrenocortical carcinoma were assessed. Adrenocortical carcinoma is destructive cancer. It originates in the cortex of the adrenal gland and generates excessively high levels of cortisol or corticosterone independent of ACTH. Thus, there is no loss of an ACTH-dependent control mechanism of the production of the corticosteroids. At this stage of my research, I had begun to develop a research program that was independent of the collaboration with Dr. Kitabchi's group, and following my passion to decode the basic principles involved in the process of ACTH-dependent steroidogenesis, I made an entry into cancer research and moved fully to the Department of Biochemistry in the University of Tennessee, Memphis. Question was, what morphological and biochemical changes accompany the transformation of the rat adrenocortical fascicular cell to the rat adrenocortical carcinoma cell? In this investigation, I first developed an isolated adrenocortical carcinoma 494 cell system that was parallel to the isolated fasciculata cell system of the adrenal cortex. The technology used in creating the two types of cell systems was identical, and so were the experimental conditions. The authenticity of the transformation of pregnenolone to corticosteroid was assessed by the techniques of double isotope labeling, tritium to C14 ratio, and co-crystallization, established prior for plant cardenolites. In collaboration with Dr. Ken Hashimoto, Chief of the Electron Microscopy Division of the Veterans Administration Hospital, ultrastructural analysis demonstrated that the isolated carcinoma cell, in a striking difference from the normal fasciculated cell, shows sparsity of lipid granules and mitochondria. Additionally, the following normal morphological features of the fasciculated cell were missing or deformed: vesicular mitochondria, excessive cellular content of lipid globules, endoplasmic secretion of lipid globules together with smooth surface endoplasmic reticulum. Biochemical analysis disclosed that in contrast to the normal, the cancer cell is unresponsive to the stimuli of ACTH, cyclic AMP, its analog dibutyl cyclic AMP, and cyclic GMP in the generation of steroidogenesis. Yet, the direct fluoroscopic and double isotopic studies with GTMC14 demonstrated the metabolic pathway from mevalonic acid to cholesterol. The formation of steroid hormone corticosterone was intact in both the normal and cancer cells. Given these facts and others that the carcinoma cell generates successively high levels of cortisol or corticosterone independent of ACTH, established that the process of carcinogenesis has created a new pathway. Therein, a lesion in the pituitary adrenal axis occurs. The lesion creates a loss of cortisol ACTH feedback loop. Thus, this in turn results in the uncontrollable excessive levels of ACTH and corticosterone in the cancer cell. In an extension to this conclusion, it was also established that the lesion resides beyond the formation of cycling nucleotides, and the phosphodiesterase activator has no role in degrading these nucleotides. With these contributions, I had begun to solidify my footing in the fields of endocrine control mechanism in steroidogenesis and the lesions accompanying it in the process of steroidogenesis. The model systems of the isolated fasciculate and endocortical carcinoma 494 used in these studies had begun to gain notoriety. Consequently, the central concept was gaining strength that ACTH is not the direct steroid hormone regulator. Other intracellular factors, such as the cyclic nucleotide, cyclic AMP, and/or cyclic GMP, may play a critical role in mediating the indirect role of ACTH. My contributions and rewards in research flowed into my personal life as well, and this will be the subject matter of my next video 6.
Thank you, Dr. Sharma, for your enlightening narration of this scientifically extremely complex problem. I am sure that all your followers are looking forward to your next episode of your scientific experience. Thank you very much.
Thank you.